Good. So let me introduce you first. So Craig Morrell is a professor and co-director of the AAB Cardiovascular, you'll tell me if I'm wrong, Research Institute at the University of Rochester School of Medicine, earned his undergraduate degree from Brown, and then went to Tufts, where he graduated, and in the School of Veterinary Medicine. Before moving to Rochester, he completed a PhD in comparative pathology fellowships at Johns Hopkins, investigating mechanisms underlying nitric oxide inhibition of platelet degranulation. And we thought he's got a slew of awards and grants and publications. He's really very well known in the field. And we thought it was really fitting that we'd start off this symposium with his presentation, starting with the megakaryocyte as the sort of the mother of all cells in the platelet stream. So Craig, it's all yours.
All right, thanks.
There and yeah.
Okay. Perfect, yeah. So I think it's also maybe fitting that I started off because I am a dual citizen. I grew up in Canada, so I've warned my family that after this I'm going to stick around a couple of days and visit some relatives. I might just stay here, so I might be calling for my family from the U.S., so we'll see how it goes over the next few days. But yeah, again, thanks for inviting me. So I think we all know here that platelets have a major role in hemostasis and thrombosis. So that's one of the major functions of platelets that I'm not going to really talk about at all today. Instead, the way my lab looks at platelets is that they're actually an immune cell that happens to have this role in thrombosis. So they really are a dual functional cell. And it's now become very well recognized and appreciated that platelets can interact directly and indirectly with immune cells at any time. So they interact directly with leukocytes through contact-dependent mechanisms, and they also dump out a boatload of immune molecules which can regulate all phases of the immune response, all the way from the acute phase as well as to innate immune cell functions. And I'll just point out another arm of our lab in one slide. So this is a recent study we had showing that not only do activated platelets drive inflammation, the resting circulating platelet is actually necessary for limiting monocyte activation. So they have this dual role. They get activated, they drive inflammation. In a resting state through this elaborate signaling mechanism, they limit monocyte activation. As a result, in conditions like sepsis, where people become thrombocytopenic, that thrombocytopenia itself might have a major role in some of the immune dysfunction. So I'll just leave that there if you're interested in that arm of some of our work. But what I thought I'd focus on today is what seem like simple questions. Where do platelets come from? And does that impact their functions? And I think it was a simple question until 2017, when Mark Looney had this paper in Nature where he showed through elaborate imaging that there's a population of megakaryocytes in the lung, and these lung megakaryocytes are actively producing platelets. But it shouldn't have been as big a surprise as it was, because if you dig through the literature over the last 100 years, there's been a large number of publications talking about these lung megakaryocytes, going back to the late 1800s. And there's actually a couple papers from the 1930s I find fascinating where they were showing that the input of platelets into the left side of the heart and the right side of the heart were I guess into the right, out in the left, were different. And then they were postulating that this lung megakaryocyte population was producing anywhere between 7 to 30% of the circulating platelets. So it's been really well known. I just think we're now revisiting it, which is a lot of things in science. Don't ignore the old literature. I guess it's a take-home point for some of the trainees, because there's a lot of great nuggets out there. So they are present in humans as well. This is just human tissue. You can see some megakaryocytes in the human lungs. They are not evenly distributed, is what I'd say. We're working through a lot of different lung sections right now. They tend to be more in the peripheral airways, and they do change in numbers based on disease. And that's something that we're kind of interested in now. But I think one of the reasons why they've been overlooked for so long is that lung megakaryocytes are largely 2N. They're about 80 to 85% of them are 2N. Very rarely do they get up to these larger ploidy. So they just, if you're looking at histology, they look just like a white blood cell on normal histology. The other thing to point out is that the bulk of them are extravascular. About 70 to 75% of them are extravascular. The ones that are making platelets seem to be intravascular, but there's this population which Mark labeled as sessile, Mark Looney, sessile extravascular population as well. So we become kind of fascinated with those megakaryocytes. So one of the things that we did to try to better define them is just the single-cell RNA-seq study from a few years ago, where we enriched myeloid cells and megas in the bone marrow and the lungs, and we found that the lung megakaryocytes actually cluster really closely to dendritic cells, right, in terms of their gene expression patterns. And why does that matter? Because, well, a dendritic cell, of course, is a professional antigen-presenting cell. And what that means is that they constitutively express MHC2, which lung megakaryocytes do, and then they can present antigen to a CD4 T cell. And here you can see by image stream that the lung megakaryocytes have markers for CD11C. This is a dendritic cell marker that they're expressing. They also have MHC class 2. In addition, your typical markers of megakaryocytes. So lung megakaryocytes are this really bizarre, I guess you'd say, kind of hybrid immune cell and megakaryocyte. So if they're going to present antigen, they have to be able to phagocytose and process pathogens. So this is just an example of bone marrow and lung megakaryocytes isolated, showing how they can phagocytose bacteria. It's a fluorescent bacteria. Lung megakaryocytes are really good at phagocytosing bacteria as well as other pathogens and substances like beads, etc. They're very phagocytic compared to a bone marrow megakaryocyte. And this was work we did in collaboration with Mark Looney, where what we did was we put this DQ of albumin, it's just basically an antigen, which isn't fluorescent until it's inside of a platelet into the lungs of until it's processed, then it becomes fluorescent, okay? So we put that into the lungs of the mouse, and they're PF4 promoter labeled megakaryocytes. But what you can see is that in vivo, in real time, lung megakaryocytes can also take up antigen and process it. So they have this ability to kind of handle antigen. So they acquire and process antigen, but do they present it then to a CD4 T cell, kind of the end stage of this potential dendritic cell-like function? So to address this, we made a MHC class 2 knockout mouse, so on the megakaryocytes. So those megakaryocytes now don't have MHC class 2, and then we could assess their functions in presenting antigen. So this is a little bit of an elaborate model, but basically we now have this megakaryocyte MHC class 2 knockout mouse. We gave it these special cells which were labeled called OT2 cells, okay? So these are T cells which recognize antigen in the context of MHC2 and it's ovalbumin. So if a cell takes in ovalbumin, processes it, and presents it in MHC2, these special T cells will recognize those and get activated, okay? So those are labeled. And then we gave the mouse an E. coli that was ova-transgenic. So if these megas are taking in the E. coli and processing it, they're also presenting ovalbumin to those OT2 T cells. As I said, it's hard to describe, but there's a lot going on. But anyway, so this is kind of the model we set up to see whether in real time in vivo the lung megakaryocytes can present antigen or not. And what we found was that if you look at that OT2 T cell activation, looking at CD25, the mice that were lacking MHC2 and megakaryocytes had reduced T cell activation and reduced T cell proliferation. It's not like it's eliminated, which you'd expect, because dendritic cells are obviously very important. It just showed in the lung itself they are having an important role, probably in that secondary activation of T cells. So that was a past study. And then after we finished that study, we started thinking more closely about this immunophenotype and how they're functionally different as well. We started to ask questions such as, where do these lung megakaryocytes come from? You do have a progenitor cell population in the lungs, which has a potential role in driving some of that megakaryocyte production. And we also started to ask questions, why are they there, right? I mean, they have this APC-like role, but clearly you can do without them. So what are some of the other reasons we have these lung megakaryocytes? So the next work I'm showing is it should be, I think it's online at JCI now, but I think the final version's out in a couple of days. So this is some newer work where one of the things that we wanted to do was set up a model where we could actually track these lung megakaryocytes. So what we did was we gave the mice this dye, CFSE, oral pharyngely. So you put it into the lungs of the mice, and it labels all the cells in the lungs. And what you can see 6 hours later, you got really strong labeling of lung megakaryocytes. You get a little background initially in the bone marrow compartment with the CFSE labeling, mostly dye overflow from the lungs, okay? So we've now labeled preferentially lung megakaryocytes. And you can look over the course of 7 days, and we don't find CFSE positive bone marrow or spleen megakaryocytes, but you do find these lung megakaryocytes. So we can label and follow the lung megakaryocytes. One of the things that struck us, though, is that there's still a significant number around after 7 days. So bone marrow megakaryocytes only live about 5 days to 6 days. These lung megakaryocytes seem to be hanging around for a long time. So we looked at later time points, and actually the first 30-day set was an accident because Alison Levita, the grad student working on this, unfortunately got COVID, and we lost track of a couple cages of mice, and we realized, hey, wait a second, we have these CFSE labeled mice that are still hanging around, and we found that after 30 days, there's still a significant portion of these labeled lung megakaryocytes are CFSE positive, and of course it declines over time, but by day 120, you still find some of these CFSE labeled megakaryocytes. So the lung megakaryocytes are really interesting and distinct in that they seem to be a subpopulation at least that are long-lived. So we want to validate this through other methods and also be able to compare that to the bone marrow megakaryocytes. So we use this biotin labeling method. So you give the mice this NHS biotin IV. It labels all the cells in the body with biotin, and then you can look later by flow whether the strep avidin binds to that cell, meaning that they've been around since the time you gave the biotin. So if you look at day 5, we see lung megakaryocytes are still well labeled with the biotin. The bone marrow and the spleen are starting to come down. Red blood cells are a positive control, right, because they circulate for up to 80 to 100 days. If you look at day 14, lung megakaryocytes still labeled. The other megakaryocytes aren't. Day 28, we still see the labeled lung megakaryocytes. So in this comparative method, using complementary techniques, again, we find these long-lived lung megakaryocytes. So this reminded us of then these tissue-resident macrophages, right? So there's macrophages in your body which live in your tissue compartments for extended periods of time. They eventually get replaced by bone marrow macrophages, but at birth at least and through the first phases of life, they're actually coming from a yolk sac, right? They're not coming from the bone marrow. So we wanted to try to get at, okay, can we better characterize these lung megakaryocytes and determine whether they're like these tissue-resident macrophages, where they have some self-proliferative potential as well. So we dug into the literature and seemed like all the cool kids studying tissue-resident macrophages do parabiosis models, so we of course did that as well. So what we did was we took a tomato reporter mouse, so all the cells in this mouse that are circulating are tomato positive. We gave it CFSE oropharyngally. We took a regular black 6 mouse, gave it CFSE oropharyngally, so now the lung cells are labeled with CFSE. We did the parabiosis, where you pair the mice for 30 days, and then we looked at the resident populations in the lungs of the mice to determine how they've turned over and whether they're persistent or not. So these are just our positive controls, and you can see the tissue-resident macrophages after 30 days are all host-derived and CFSE positive. So our control is working, okay? And if you look at the lung megakaryocytes, you can see after 30 days, about 50% of them are host-derived CFSE negative. So they've been turned over from a bone marrow, host bone marrow source. You also see some of these megakaryocytes which are partner-derived CFSE negative. So those have come from the partner. So there is a circulating source that's repopulating the lung megakaryocytes. We also in both mice see CFSE positive host-derived megakaryocytes, which have just been there for this 30-month period. So again, it shows that they're coming from a circulating source, but there is the number of them are tissue resident over a longer period of time. So to kind of rule out that they're a yolk sac derived, that they're not HSC derived, we use this reporter mouse model, basically MDS1 Cre-ERT. So if you treat these mice with tamoxifen, any cell that's derived from an HSC after you give the tamoxifen will be tomato positive, okay? And if you look here, if you give the tamoxifen at 3 weeks of age and you look at 7 weeks of age or 8 weeks and look at 14, you can see that the vast majority of these lung megakaryocytes are tomato positive. So they have come from an HSC. So we don't think they're yolk sac derived, we think they are HSC derived. And just as your control is microglia, which of course are yolk sac derived, and none of those are tomato positive. So they're coming from the bone marrow, okay? And just to make things more complex, of course, there's two ways that you can get megakaryocytes in the bone marrow. One is this regular kind of constitutive pathway where it goes from an HSC to a short-term HSC to an MPP and then down to your megakaryocytes, right? So that's kind of the standard textbook way that we describe megakaryopoiesis. Oops. But there's another way that you can get megakaryocytes. Actually, I'll go over to here, which is described as a non-canonical or direct pathway, which is increased. This pathway is increased during times of inflammation as well. And what happens is there's this direct pathway from the HSC to the MKP. So it skips the MPP phase, right? So it's kind of this direct pathway to get megakaryocytes quickly at times of stress. And there's a reporter mouse you can use to track these, and it's this flick switch mouse. Basically, when it goes to the MPP stage, it expresses FLIC2, which is also called FLT3. Gets a little confusing. But when it does that, it cleaves out the tomato reporter, so everything downstream is now GFP positive. So if a megakaryocytes come through the constitutive pathway, it's GFP positive. If it's come through this direct pathway, it's still tomato positive. And if you look in the lungs of these mice, about 90% of them are through this direct pathway. So these lung megakaryocytes are coming through this alternative non-canonical pathway, whatever you want to call it, and then populating the lung. So they're even derived through a different pathway, which might give them some of these different characteristics. And I'll point out a really interesting paper that was in Cell from Camilla Forsberg's lab in May, showing that this non-canonical pathway is actually amplified during age. So at least as mice are aging, there's more of those megas and platelets in the bone marrow itself as well. Okay, so lung megakaryocytes are HSC dependent, but FLT3 independent, and they're long-lived compared to bone marrow megakaryocytes. But do they make platelets, and how many platelets do they make? And I'll point out that the how many is kind of this controversial area where Mark's paper indicated that it might be up to 50%, which became a big touchstone point in his paper for how many platelets are coming from the lung. But we used our CFSE labeling methods to try to get at this. So basically, if you give them CFSE oropharyngally, and at the same time you give them an IV platelet labeling antibody, and then over time you can do flow cytometry. So the CFSE positive platelets that are CD42C negative are new platelets coming from the lung in this quadrant. So if we do that, what we can see is that lung megakaryocytes are making platelets in real time. If you normalize this to the total number of megakaryocytes in the lung, we estimate about 10%, 7 to 10% at any point of your circulating platelets are coming from the lung. I do have to point out a caveat that over time this is going to be labeling those tissue resident ones, which might be making platelets, not the ones which might be coming from the lungs. So the caveat is this is the tissue resident megakaryocytes contribution, I guess is the best way to phrase it. So if you look at those platelets and how they respond normally to thrombin, they respond normally to thromboxane, but they respond more to these TLR agonists, right? So they've increased activation to TLR2s and TLR7s. They also, if you do RNA-seq on these platelets, they express more genes, the CFSE positive versus negative by flow sorting. They express more genes related to immune molecules as well. So they're immune differentiated, the platelets, that respond to TLR ligands. So do they make more platelets following an acute depletion? So on demand, can they make more? So we use this DTR mouse model where basically, if you treat this, it's a PF4-Cre DTR expression. So any cell which expresses PF4 is expressing the diphtheria toxin receptor. You treat those mice with diphtheria toxin, and it eliminates platelets and megakaryocytes, which then rebound, okay? And you can see that after you give DT, there's a decline in bone marrow and lung megas, which then are restored. And at this time of increased demands, we see more CFSE-derived platelets. So with increased demands, the lung megas are making more platelets. And we can do this in an infection model as well. One of the models we use a lot is this malaria model. So it's an uncomplicated malaria model. If you give it malaria, it becomes thrombocytopenic. You can see here over about 3 weeks, they clear the infection by day 4, and everything goes back to normal. And if you look at the megakaryocytes, early on, bone marrow megakaryocytes in this infection go down, whereas lung megakaryocytes go up. And if you look at the contribution of platelets, these CFSE positive platelets, in infection where there's thrombocytopenia, the lung megakaryocytes are now making more platelets. So they might be this reservoir reserved to make more platelets upon demand, particularly in these inflammatory states. And kind of as a last a bit of data on what they're doing, one of the things we looked at is these resident megas, which are sitting there for a long period of time, can they be called later to make platelets? So Allison gave the mice CFSE oropharyngally, left them for 3 weeks, did nothing to them, and then infected them and looked a week later at whether they were making platelets or not. And you can see at baseline, those kind of resident hanging around megas aren't really making platelets. After infection, they are. So they can be called to make platelets after a long period of time of sitting there waiting for some action. So our best characterization of these cells is that they're FLT3 negative alternative pathway derived, that they make more platelets with increased demand, and they're more immune stimuli responsive. And that's kind of what we're thinking they're doing. And now some of the questions we're asking is how are they getting there? So if you do flow sorting of tomato positive, GFP positive, bone marrow megakaryocytes, so these flick switch mice megakaryocytes, you can see the ones which are going to the lung, the tomato positive ones, and the ones which are staying in the bone marrow. They have very different gene expression patterns, and it looks like those non-canonical pathway ones express a lot of chemokine receptors. So we're looking at whether that's one of the drivers of how they're getting to the lung. We're also looking at some of the regulations. So one of the things we stumbled upon is that bone marrow megakaryocytes express this transcription factor MIB. Lung megakaryocytes don't. During infection, bone marrow megakaryocyte MIB goes down. That's a time when the bone marrow megakaryocytes also have increased immune molecule expression. So is MIB kind of a master regulator of this immune differentiation pathways is another question we're starting to get into now. So with that, I'd like to thank members of my lab and point out that the bulk of the work that was done was by Alison Levita. So she's a outstanding MD, PhD student who's now back in the salt mines of the MD world, but she did the bulk of the work that was talked about today, as well as our collaborators, Jim Pallas and Kathleen McGrath, who are just amazing hematopoiesis experts who we rely on a lot. So I think I'm relatively on time. Thank you. Yeah.
So we're open for questions. Please use the mic. So we have Mike Runner. She really does run. We only have one mic runner. We have another. Okay, good. Okay, so Geraldine, go.
Thank you so much. That was a really interesting talk. I was just curious if anybody or yourself has looked at the expression of immunoproteasomes, either in the bone marrow or the lung-derived megakaryocytes or in their platelet populations.
I'm sorry, the immuno what though?
Immunoproteasomes?
No, not at all. That's a good question. I mean, so they do have a proteasome, right? And so we know if you give platelets DQ of albumin, for example, they'll become fluorescent because they can cleave and process those antigens, but beyond that, not at all. I mean, they have TAP proteins, right? They have all these proteins to load MHC1 with antigen as well. But beyond just saying they're there, that's kind of it, yeah.
All right, thank you.
Yeah.
Great
Yeah.
talk, Craig. I wonder if you could look at the lung megakaryocyte-derived platelet. Do they have more immune signatures on them? Or would you
I kind of breezed through it, but we have done flow sorting of CFSE positive and negative megakaryocytes and did RNA-seq on those, and they do have very different gene expression patterns which phenocopy the lung RNA-seq, right, or the bone marrow megakaryocyte RNA-seq. So they do. The challenge when you're trying to do flow cytometry for low expressed molecules on a small cell is you get these shifts in fluorescence, but they're not enough where you feel comfortable saying this is a marker of that cell, right? So if they were bigger, then maybe it'd be easier because you get more molecule expression, you see bigger shifts. But that's one of the challenges right now is trying to find something that's a reliable surface marker versus our CFSE, and we're going through a long list of molecules, yeah.
Michelle.
Hi there. Really interesting talk. Did I understand correctly when you said that the platelets were more receptive to TLR2 stimulus than thrombin?
Yeah, so what I would say is that bone marrow and lung-derived platelets respond very similarly to thrombin, but the lung-derived megakaryocytes are more responsive to TLR2 than bone marrow. I wouldn't say thrombin's still the best stimulus, right, if you kind of do a comparison, but the lung-derived ones will respond more strongly than the bone marrow-derived ones to the TLR agonist. Yeah.
Do they express higher numbers of the TLRs?
Yeah, on an RNA level they do, and again, this is one of those things where we see those small shifts by flow cytometry that indicate that they're probably that they're expressing more on the surface as well. But by an RNA level, they definitely do. You know, we do. I think the best way to do it is once we flow sort them, doing immunoblots, but that just requires a lot of time and and detail. Yeah.
Thank you.
Okay, over here.
Over there,
Okay.
yeah.
So really interesting and fascinating studies. Your studies are all murine.
Yeah.
Do you have any human equivalent?
Yeah, so just the immunostains, right, from our lung map. We have a group that does this lung map where we have access to a lot of tissues, and thankfully they've included some of our markers of interest. So the one area that I think would be really cool, is people right after they get a lung transplant, right? Because you could then look at different haplotype markers. They just don't do lung transplants right now in Rochester, so we're trying to coordinate with some Cleveland Clinic people where they're doing a lot of lung transplants, and I think that would be kind of interesting. And I think if we can nail down a reliable surface marker as well, that would make a world of difference. And that's one of the reasons why, you know, like Emily was kind of indicating, to go through that list and see, can we see reliable shifts as surface markers? Yeah. That's what's missing to really make it translational. Yeah, definitely.
Craig, with these tissue resident sort of story, is there any role for these megakaryocytes in COVID?
Yeah, so that's a great question, and it comes up a lot. I don't know right now, right? So there's been some really interesting studies showing that you have increased circulating megakaryocytes following COVID, right? And that those circulating megakaryocytes in those studies are also CCR7 high, which makes us kind of think that they look like these ones which might go to the lungs. In terms of driving the lung pathology, I just don't know. We don't have a reliable BSL-3 facility, and in hindsight, it's probably good because that we didn't get down the COVID road, but that's, so I can't answer the question right now other than to say likely, but I mean, the other model I think which would be more fruitful is influenza, and we've got lots of colleagues doing influenza, and we're kind of gearing up to do some of those, yeah.
And just drawing on your comparative biology background, are there other species like zebrafish or something like that have a similar mechanism?
That's a good question. I don't know, yeah. I don't know. I really don't know. Yeah. And, you know, and some of the evolutionary pressures for this is something that I think about a lot, and I don't have a clear answer on it. I mean, in the back of my mind, I think they might also be a way. So we suck in, you know, antigens all the time, and it's a way to kind of see low levels of antigen, more for immune tolerance as well, because if our MHC2 knockout megakaryocytes, at baseline, those OT2 T cells actually proliferate a little bit more. They don't increase with infection, which leads me to believe there might be a tolerance role, but I don't, proving tolerance is harder than activation, so yes, that's down the road, yeah.
Any others? Thanks so much.
All right, thanks.